Welcome, everyone. Thank you for coming. And we do have a virtual option, and we have people online, so we're going to make sure we use the microphones so that they can hear us. I'm here for the housekeeping portion. So welcome to the 2025 David A. Wilson Innovations in Medicine Distinguished Lecture Series. We are going to be really sort of excited to have our guests talk about digital twin. Did I go forward? Yes. And then AI blood-driven analysis. So we have the Dr. Wangs here with us. Yes, they do get each other's email, and they are very used to talking about that. They know each other's secrets. And I'm really thrilled because just, in talking to them over the past few weeks and learning about their work has been really, really interesting. And I feel very positive for the future. So this is CME, so please make sure that we're going to give you the code in a moment. So to have CME, we have to say the learning objectives. So for the first speaker, we have for the digital twin, we're going to understand the concept of what digital twins in healthcare really is. We're going to explore applications in precision medicine and examine the role of AI in computational modeling in healthcare and medicine. And then our second talk will be on AI-driven blood analysis. So we're going to gain a basic understanding of the complexity of human blood proteome. Thank you, it's been a while from med school. Learn the principles of solid phase extraction and protein enrichment and understand the fundamental concepts of mass spectrometry data analysis for proteins. So go ahead and text the code for CME. This will be available for some time. And we'll also play this at the end, so if you hadn't have a time. This also is going to provide the Academy and educators credit. So for those of you who aren't sure what the Academy of Educators are, it's a School of Medicine, Greenville here, establishment of a community to honor, support, and enhance our faculty in their teaching, mentoring, and scholarly activities. And so if you want some credit for AOE attendance, this is the scan. So also at the end. Great, and so I'm thrilled to have Dean McGilvery come up and kick off this wonderful evening.
Dr. McGregor. So for those of you I haven't met, I'm Dr. Phyllis McGilvery. I'm the Dean of the School of Medicine here in Greenville, and it is my honor to invite you all to this talk, which I'm super excited about. I would like to thank you for joining us tonight. Thank you for Bob and Teresa Wilson for being here with us, and for who are trustees of the Sargent Foundation, and for putting this lectureship on for us. Additionally, thank you to Prisma Health, School of Medicine, Greenville leaders, faculty in the room, everyone for being here this evening. As many of you know, we have a new mission statement, which is Educate, Innovate, Serve, where lifestyle is medicine. And this is another piece of that educate and innovate space. And we're looking forward to hearing all about the topics that we're here today. We are very, very grateful that our medical school has vital, visionary partners like the Wilson family joining us in building a future of healthcare. And this is another example of that with these innovative speakers tonight. The Sargent Foundation's long-running relationship with Prisma Health and the School of Medicine Greenville, their visionary gifts have fueled a wide variety of opportunities, including the Martha Ann and Hugh Clark Endowed Scholarship, the SIM Center in Innovations, lectureships such as this one, our Student Summer Research Program, the Anatomy Lab, our new mobile teaching kitchen, and the Sargent Foundation's Dean Suite. So many wonderful and special gifts through the generosity of this family, and I thank you so much for that. So I'm going to talk a little bit about, Dr. Wilson. So David Wilson was the first board-certified general surgeon in Greenville. He helped secure the first accreditation of surgical training at Greenville Hospital Health System, now Prisma Health. Dr. Wilson instituted a formal training program for surgery here and conducted groundbreaking research on tuberculosis. His family, along with the Sargent Foundation, established the lectureship series in celebration of this innovative work, including his research and surgical treatment of tuberculosis. And in the memory of his outstanding leadership in South Carolina, this annual lecture series was founded to bring prominent speakers to address and consult with our school's community on topics of high importance to both healthcare and leadership. And so I would love to invite Dr. Wilson, Dr. Bob Wilson, up to share some sentiments about his father with us.
Thank you, Phyllis, for giving me an opportunity to just speak briefly. I certainly appreciate y'all sponsoring this lecture series. My father came to Greenville 80 years ago, and I appreciate the lecture series honoring my father. He did his work on tuberculosis, surgical treatment of tuberculosis, and I'm sure you're aware that probably about 1 million people a year still die of tuberculosis. So it's still a scourge in the third world countries. And who knows, maybe they'll institute some surgical treatments for TB again, since the anti-tubercular drugs are not working well, particularly in India. Not only is this an opportunity to recognize my father, but also to just make some observations about how medicine has changed in the last 80 years in Greenville. When he came here, all doctors were autonomous. They worked for themselves, and about 99% of the doctors were men. There were very few women. The doctors would all come in a similar fashion and set up an office and hope over the years they could get enough patients to make a living. At that time, a lot of the doctors were not even university trained. They would train like an apprentice system. So some of them were well trained, but a lot of them got anecdotal training and really did not have a very broad scope of practice. At the time my dad came into town, St. Francis Hospital, which had about 250 beds, was run by 2 administrators, the nuns, and it seemed to run very smoothly. And the Greenville hospital system only had about 5 or 6 administrators. So there's been a lot of big changes in medicine, not only in the actual practice of medicine, but the delivery of medicine, particularly with the hospitals. And I just think it's an opportunity to reflect and think what medicine will be like 25 years from now, particularly with AI, because of the revolutionary changes it's already making in some professions. And I certainly look forward to their comments today.
Wonderful, great. So we are excited to start. We're going to start with Chi Wang, who's a professor of mathematics at the College of Arts and Sciences at University of South Carolina. He's going to discuss digital twin for health. Think of a flight simulator, but for your body, or for your tissue, or for your organ, right? So fascinating, so revolutionary. His research interests include modeling and computation of complex systems in materials and life science, development of efficient numerical algorithms for partial differential equations, data science, machine learning applications in materials and life science. He's currently developing an individualized, multi-scale, multimodal digital twin framework for cancer patients to be used in optimizing the patient treatment pathway. He is especially interested in developing cutting-edge deep learning models to approximate complex biomedical systems of spatial temporal resolution. He's published over 200 peer-reviewed journal papers and has been continuously funded by the federal funding agencies. So please welcome Dr. Chi Wang.
Dr. Mike Greger, and also thanks for the Wilson's family's sponsorship, and Dr. Dean Mike Gregory. And also want to thank Dr. Mary Rojek, show me around this afternoon. So it's my pleasure to join the faculty and staff here to talk about my work. The work is very recent because before the COVID, in 2019, DOE and the National Cancer Institute issued a so-called Ideas Lab invitation to the worldwide research community. And they want to find out how they can frame the concept of digital twin for health. So they want to organize a Ideas Lab. I don't know any one of you heard about this concept, Ideas Lab. Basically, you bring experts from all walks of life and then try to brainstorm within 5 days in a week. And then by the end of the week, you need to write a proposal to compete for like $100,000 seed grant. So I submitted my application and told them my expertise in modeling, computation, and with especially my extensive knowledge of non-equilibrium thermodynamics. And then I was selected. And then COVID hit. And we did not have a face-to-face meeting, but instead we have online workshop for 1 week, organized by a workshop company based in UK. So we came together knowing nobody in the group, 35 people, several from Europe, several from Canada, and most from USA. And then we try to figure out what we mean by digital twin for health. Okay? And you can imagine, the idea was very rough. We even did not have any convergence by the end of the 5th day, but at least we have time to write a 3-page proposal and then compete for the $100,000 grant from DOE and NIH. And we got it. Actually, 6 teams submitted, 5 teams got funded. So the percentage is pretty high. And then we started this project. So we try to refine this. And I wonder, how do I flip this? Over here. This is where we begin. So we try to frame digital twin for health. And actually, if you look at this concept, the concept existed for a very, very long time. You think about the aircraft maintenance, Boeing or militaries. They cannot climb on the aircraft and examine all the fractures. What they do is they set up a database, and then they use computer simulation to try to estimate the fatigue or potential fractures, and that's the prototype of digital twin for aircraft. And only until 2002, one guy in aircraft industry, he used the word digital twin. And then this word came about. So now this word is spreading around in all kinds of fields. So basically, this is a virtual representation of a physical system, whatever that is. Aircraft, you want to do a urban design, a city, you want to design a car, you want to design a ship, anything, you name it. Then you can create a replicate digitally. And the good thing about this is you can use whatever existing data to create it. And the good thing about the digital replicate versus physical one is you can go back and forth. You can make predictions, you can also trace back. For our human being, we only live at our being, right? We cannot go backward. We cannot go even into the future. But digital version, you can go either way. So that's the one of the virtue of this digital twin. So for us to create a digital twin, we need all the historical data. And with that, we can create some kind of an inference capability. So we call that predictability. We want to make predictions, okay? And if we apply this concept to healthcare, in particular in medicine, I think there's a huge market, huge potential for this. So that's why we want to use so-called multi-scale. You look at the data we collect for patients, you can collect data on weekly, monthly, even yearly, but you also have the data collected when patient is in ICUs. You can collect that even in a minute or even second these days, right? So we have this multi-scale, and also we have multi-model. Imaging, physiological data, genomic data, all kinds of other forms of data you can collect. And therefore, if you can integrate all this into one virtual entity and then create a copy of this disease or subsystem of the human, or ultimately the entire human body, that would be great, right? So that's the ultimate goal. So we're going to shoot for. But at this point, we have to be realistic. Let's focus on one system at a time, whatever system we can put our hands on. Right? So the use of the digital twin, actually, there's a lot. So I'm going to mention that here. If you look at the digital twin applications, potential application in healthcare, there are a lot. We list a few of them. For example, personalized medicine. You can always create, for single patient, you can use all the collected data in the past to create for certain disease. For example, diabetes or heart problem, even cancers, right? So let's focus on this specific system. Don't focus on a human, because human, that's our ultimate goal, maybe 10, 20 years from now. But now we have to build from scratch. And personalized medicine is possible through digital twin. So for example, now we're working on cancer, certain type of cancer, non-small cell lung cancer. We can collect all the data from the ERM, medical record, and also hopefully we can have collaborator collect some newer data, because newer data will be much more cleaner, more complete. And then we can build this dynamical model, okay? And the other one is real-time health monitoring. I mean, even you're a healthy person, you would like to have some virtual replicate of yourself, right? And for example, you are concerned with your heart, or you're concerned with your glucose levels. If there are some device available there, which can really track everything happened in the past, and make some predictions for short time in the future, I guess you would like to have that kind of devices as well. So digital twin can fill the gap here. And also predictive diagnosis. This probably is the most useful digital twin application in medicine. Based on all the information you have, experienced doctor, can make diagnosis and prognosis, right? I guess prognosis is the most important for patient. They already want to say, okay, with this treatment, what would be the outcome, right? So if you can have some model to make that prediction, and maybe just short time, that'll be very valuable, not only to the patient, but also to the doctor. I remember, my wife had a problem called a PBC many years ago in 1995. At that time, a few models available, they said, okay, you have survival time up to 15 years. Okay, that was in 1995, so 30 years from now. She's feeling very well, at least with that PBC part. And then now when we talk to the medical doctor, they say, oh, PBC is no longer a problem. Okay, so even though that model is not that accurate, but at least have a model is better than without a model. Okay. And there are several others, like surgical planning. This is very obvious. If you can have imaging, everything integrated into a digital twin, and when the doctor designs sophisticated surgical procedure, that can really help, okay? And most importantly, I think when we talk to people with applications of digital twin, is drug testing. They think pharmaceutical company will really love this, Because so far, you do whatever drug testing, you begin with 300 patients. By the end of the trial period, maybe 50% or less are left, right? People just keeping drop out, either because they become, ineligible, or they just, tired of this process, they drop out. So digital twin can really, if we can have a digital twin put into the drug testing instead of a real human, they won't drop out, right? Unless, unless some, electricity is gone or whatever, right? Otherwise, they are going to stay. So this is another application. There are several others. I don't think I have time to run down the list. So there's many places you can design digital twin, develop it, and then apply to the real-world settings. So here is a schematic, and maybe you can see a little bit better. So you can have clinical trials, you can have wellness, you can even have a hospital management. You can design digital twins to do that. And this is a little bit more details. So what is a digital twin? Well, digital twin need to build using whatever available data you can collect from the hospital or even from the databases, because sometimes the hospital data may not be sufficient. For example, you only have 300 data, for 300 patients. But for machine learning, like ChatGPT, when they train that, they use almost all information available from the internet. So how do we do that? Well, there are several technical ways we can do. For example, you can look for similar patients using so-called similarity analysis, then you put them into the patient pool, or you can create synthetic data. This is a new technology where we are experimenting now. You can add more fake data, but they are created from the same probability distribution from the patient. Okay? So with all these data types, then we can build a digital twin. And then once you have the digital twin for physicians, you can use this to look for so-called treatment path, for many, many different diseases. I know there are multiple ways you can treat them, right? So you can, using this medicine or that medicine, and even for same type of disease, then you may have slightly different outcome because each patient is different. So care path design, that's most of the time is a physician's work, but now probably the digital twin can help you. I'm not going to say digital twin will replace you, but definitely will help you, okay? And also pathway optimization. This is the most important thing on the mind of patient. Anytime if you give patient a treatment plan, and then they would like to say this is the optimal plan, right? But how you can make sure this is optimal or non-optimal, I think using AI and optimization algorithm can really help. Of course, experienced physician is doing that already because that's what they are trained for, right? And also based on that, then we can have, when we build this process, we need a lot of technical stuff. For example, data curation, data standardization, and the modeling and the simulations. So those parts normally is done by people like in computer science, in statistics, in mathematics, or even in more quantitative, aspect of medicine. I don't know, maybe we call that medical scientists, right? So people were trained with more knowledge in physics and mathematics. So once you have those, and then we can design this, and then once this is designed, then you can put into a test. So this is my vision. Probably some words I hope is not offending to the medical doctors. So I think a digital for healthcare, digital twin for healthcare will be an intelligent aid to physicians. Okay? And also it can help physicians to make diagnosis and the treatment decisions. And also can make prognosis and estimate for the treatment trajectories, because you may have different paths going forward. And this may really tell you which one maybe is the best at that moment, okay? Maybe this will be changed over time. And also this will help physician to design the optimal treatment pathway. I think that's very important, okay? And also the fourth item maybe is a little bit of funding. I think when this becomes mature, it will be better, the decision will be better than an average physician. Okay, this is, up to debate. But this tool will never replace the role of a physician, because ultimately will be the physician to make the treatment decision, will make it. But this will be a intelligent tool. So that's my vision for this. Now here is a design of what we are doing. So for example, for cancer patient, we have all kinds of data from the patient, right? Starting from clinical, physiological data, from blood tests, from genetic data, cancer markers, biopsies, and also the doctors' cancer staging, all this data. So we're going to use all this data to design different modules for the patient. For example, we can have physiological module, that's what we're working on. We can also look into the morphological evolutional module, where you have a solid tumor and how this tumor will evolve with the treatment and with the progressions. All this can be monitored. And also we can put into like a drug toxicity assessment module. And even we can put into dosage planning modules in the end. So far we're working on the top two in the last couple years. This is just the grand, scheme for us to design this digital twin for cancer. And for cancer treatment, this is the cancer treatment for non-small cell lung cancer. You have so many different paths, right? And how do we find, for any particular patient, how do we find the right path which is the best for this patient? So that's where the digital twin can be put into use. Okay? And this is another way, how do we really, I don't want to go into details, so this is in our proposal. How do we really use digital twin to do the forward feeding and the backward feeding so that we can utilize this to incorporate it into the doctor's diagnosis and the prognosis process. All right, so the message for this project is a digital twin can really provide critical, timely, and responsive insights for individual cancer patients. So that's what we expect. AI plus mechanics modeling plus high-performance computing will enable one to move from observational type of treatment to prediction and more accurate decision-making. And of course, understanding cancer biology with the medical knowledge and also patient care trajectory is the key. But so far, we have some bottleneck. The bottleneck is the data. So data, a lot of data are not sufficient. Give you 100 data compared to the training of ChatGPT, that's like a 1 versus 1 zillion, okay? So it's too small. So here are two of the current projects I just want to mention. The first one is we are developing a prognostic model for ICU patients based on the routine biochemical indicators. So the idea is when the patient is very, very ill and is susceptible to sepsis, and then the patient is in ICU, how can we help physicians to prepare their decision within hours, okay? We're not talking about a chronic problem where we can prepare that for weeks or months, okay? And here are some models we use to make the data more complete, because all the data we have, regardless from the database or from doctors' own repository, there will be always missing entries. That's always norm. Sometimes 10%, 20%, sometimes even 40 or 50%, okay? So now we develop a model where we can fill in those missing places so that the data after the fill-in is still reasonable. Of course, this is under some mathematical assumptions. So here are the biochemical markers we are testing, about 40 of them. So we can categorize them into 4 categories. So blood counts, liver and the kidney, function, and coagulational function, and the cardiac injury markers. And also here are some missing, percentage of those. And I just want to show you the results. So once we using what we call the diffusion model, which is the best so far, and our error is about 0.25%. Okay, that's very good. Okay? So I don't have time to go through this. And those are the results where you can look at the red dots were the ground truth data, and the other curves and the gray areas, those are the predicted area, the data. And also we can pull the result together using the so-called ensemble method. And I don't know if you know, when people make the weather forecast, you never have any accurate weather forecast, okay? And then they find out over the last 100 years, the best method is the ensemble method. In other words, you assemble all the results from around the world, then you average them out, and that gives you the most accurate result. Now, we do the same thing here for medicine. We get the results from different models, then we average them out using different ways. Then turns out this gives you the best results, okay? So here is what we find out. So if we look at all those biomarkers, and of course this, you look at the last row, this tell you the accuracy is about 82%, okay? Among the 300 patients data plus 200 we created using so-called diffusion model, okay? And there are several other markers, it's roughly around 80%. So I was told by my collaborator, 80% clinically is acceptable. Of course, we would like to push this number higher, but that will require the patient data to be, maybe 1,000 or 10,000 at that level. We can push that higher, okay? And the AUROC, that's another, I guess some of you may be familiar with. That marker right now is 88%. So it's not bad, at least clinically acceptable, okay? And also we can inject some interpretability to this. So using a so-called XAI, interpretable artificial intelligence, we can identify what marker is most important to the decision the AI model makes. So in this case, for the negative result, we have the ALB is the first, is the most important. And second one, I guess, is RBC, the red blood cell count. And third one is the plaque. And for the positive result, it's still the three, but their order is slightly rearranged. So this gives the practitioner at least a tool to see which one is most important when they make decisions, when they read the data. Okay. So I guess my last one is another model. We try to make glucose predictions. So this one is on single patient because we have a collaborator who can collect data every other minute when the patient is on the machine. So now we want to try our AI models. Okay? And that patient is susceptible of sepsis again in the ICU. Right? So let me just show you the result. So we tested 5 different models, and all these bars measures the so-called error. Okay? The lowest one is the best. So now we have one model which gives the lowest bar, which is roughly about 3% error. Okay. And then we also tested the ChatGPT. So we said, well, ChatGPT is readily available. A lot of doctors, a lot of people are using that. Why don't we try the ChatGPT? So we designed certain intelligent questions and then gave all the data to the ChatGPT and then ask ChatGPT to sort them out. So we do this in many different times. So for example, we ask same question 10 times. We got 10 different answers, then 20 times, 30 times, right? And then we average those answers. So here are the results. So we see. Yeah. So the first one is 15 answers average. The second one is 30 answers average. The last one is 60. So I guess, yeah, so the boldface, give you the smallest numbers in each category. Okay. So anyway, so this tells you the ChatGPT is usable, but it's not the best, it's not the worst. It's in the middle. But we do have good machine learning models which can give you very nice result. At least in this case is better than 80%. Okay. So in summary, digital twin for health is one of the emerging field. We also call that the disruptive technology, hopefully that is. And this one requires people work together from medicine, from IT, from modeling, from mathematics. And it could be the next, darling for Wall Street, because every 10 years they are looking for the growth areas, right? So DT could be one of them. Of course, we hope that's the case. And also there are several other issues I have not mentioned, for example, ethical issues, societal issues, and the legal issues. All those are also associated with the digital twin, and hopefully someone will fill in the gap to address that issues. But I think for the medical doctors, digital twin gave you a toy you can play around at the beginning, and then you can judge whether or not it's useful. Because in the end, it's going to be the medical doctor, professional, and also the patient will be the user for this technology. Thank you very much.
Questions till the end. So let's bring up our other speaker. Thanks. So next we'd like to welcome Dr. Chen Wang, who is the co-leader of the Thrust 1 of the SC ADAPT program. I'm sure we're going to hear about that. He received his BS and his PhD degrees in chemistry from Beijing, China. And after postdoctoral experiences at the University of Lausanne in Switzerland and the Scripps Research Institute, he started as an assistant professor at the University of South Carolina in 2003. He then was promoted to associate professor, full professor, and is currently the Carolina Distinguished Professor of Chemistry. The overall research objective of Dr. Wang's laboratory focuses on using chemical biology tools to probe intracellular activities and the development of hierarchical structured nanomaterials to study the cooperative response of cells to extracellular matrix. This is why I'm an MD. He has supervised more than 100 doctoral students, postdoctoral fellows, delivered over 160 invited talks, and published over 300 publications in peer-reviewed journals. Please welcome Dr. Wang.
Thank you so much. Thank you. This is such an honor, and it makes me very nervous because Qi and me, we know each other for many, many years, and we are brothers, by the way. But I think the first time we really give research talk back to back. So, and then he, as you see, he already set a very high bar, right? So, and also it's very, it's a great pleasure to come here to Greenville, a lovely city, and then to come to the school. And thank you, the Dean McGilvray, and then thank you, Dr. McGregor, to put this together, right? And then Mary, and then where is Kathleen, right? For to really bring me here. So, where to start, right? So we had a very nice, conversation with the Bob and then Teresa, before the. Okay, no, here we go. So that's the talk. So really, I want to do something is still under the same theme of the AI, but instead of digital twin, I want to bring two other aspects and just try to really see whether our research can stimulate some kind of continued discussion and maybe can encourage the the further collaborations. So how to make some the conflict interest but anyway, so one thing I have to say so I'm a chemist, but my family my wife is a physician. So I really appreciate everyone here, and I know how busy you are, and you can come here to listen to the talk, right? It's really difficult. I talked to Bob and Teresa during our conversation. My daughter Florence, so the older one, graduate and then try to use the data science and then to really provide some kind of solution for biomedical care. After 1.5 years struggling, and then she is thinking maybe she should go back to medical school. So we senior doctor, right, My son, the second one, Leon, I think is thinking to go to medical school also. Will go to potentially join the MD PhD program at the MUSC, right? So really, I'm surrounded by all the doctors. So one thing very important, I think, we know, as the research, right, in the institution, the 3 pillars of the support are very important. The federal funding, the private sector, and then the generous donors. That's another thing that makes me really honored to come here because this lectureship is funded by Dr. Wilson, and then also with the Sargent Foundation, right, together. But more importantly, I talked to Bob and Teresa and really impressed about how much they really involve in the development, the research of the medical school in Greenville and the big South Carolina area. I think we really need someone like the Wilson family to provide more support in order to really promote the cutting edge research and then to really move us to next era. So I want to really shift gear a little bit to one specific question, and I hope it can give you some kind of idea why we are interested in such kind of question. So I just want to talk about one thing, because as a doctor, we all know the blood analysis and also some other body fluid analysis is very important. But there are 4 very difficult challenges we are facing every day, especially if we move to the individual or the really quick analysis of the patient. First one is, I'll use this screen here, right? Just make sure everyone. So the first one is how we can really capture the transient process, the metastable protein signals during the shaping and analysis. For most proteins, right, not only protein itself, but also we call it post-translational modification is very important. But many of them, like phosphorylation, or glycosylation, simulation, or other PMT we call it, right, is really unstable. But those kind of modifications are critical for the diagnostic analysis. So how we can capture them. Second one, right, how to really give the individual analysis in a timely manner here. That is important for many, many different, the diagnosis or prognosis, the practice, especially in MSEO, in the urgent care, right? How can get, in this hour or in a few hours? I think everyone experienced that one, right? My wife always complain about, when she has patients, she has to send sample to LabCorp, right? And after 40 hours, they lost sample, right? I call them again, they say, I'm sorry, send me again, right? Take a week to get the data back. But that's something for doctor is a nightmare because, we are working with MSU doctors. They said, we need the information in 6 hours, 8 hours top. After 8 hours, you give them the data, I can only put in a note. There's nothing to do with my treatment now, right? How can deal with that? I think there's something, can we find a solution here? There are another thing here for the really a lot of data in the blood, for example, how you can really get the data from or the information from those kind of high or remove the high abundant protein interference. So I'll talk a little bit on that one and why that's a challenge. And finally, how we can make sure, the information we get, from thousands of proteins and metabolites, they are relevant to the doctor, really the final consideration. So that go to, the 2 solutions we are thinking about. So when we talk about AI, not only the data here. So I think for the science point of view or research point of view, we talk about 2 things. One is the data, right? How to use the data to do the analysis to help us to assess the situation. Second one is automation, right? And automation is part of the AI. How can you use the current, the tool to make something automatic, to reduce the human error, and how to really make faster and then accurate. So I'll go to that one later, but let me just talk about one thing here. Is something, why I talk about, the blood analysis is difficult. It's something we are facing every day. So what I show here is the blood composition. You can see about the 40% of blood proteins is albumin. the concentration of albumin is about 30 to 50 milligrams per milliliter. Just remember the number here, right? 30 to 50 milligrams per milliliter. But the protein really important to many, the individual patients. They are here. This part, we call it, the low abundant proteins. Let me see. It's really, it's about 1% of the protein. They are critical. All the others, about 20 proteins, they occupied 99% of all proteins, right? It's just crazy, right? But in this part, you have about something like thousands or couple thousand proteins. They are here. So let me show one thing here. Typically, let's do a diagnosis, like the oral cancer biomarkers. So there are a few of them here, but for all of them, they are the low abundant proteins. The concentration we talk about from micrograms per milliliter to sometimes a picogram per milliliter. Think about the albumin is 30 to 50 milligrams. So we are talking about a 10 to the 6, 9-fold difference here. So that really interferes with all the analysis we can do if we don't remove them or we don't really take the sample or the signal out. Similarly, she talked about sepsis, right? The biomarkers here, right? Typical biomarkers is nanograms, low nanograms, right? And to picogram level is really, 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 really low. How to do that, right? That's one challenge, we are facing. So typically how they do it in clinic or if you send to LabCorp Quest, right? They call it SPE here. So use a solid phase extraction, right? You have the blood, do filtration here, and you remove the serum or plasma depending on the applications. Then you mix with some kind of column. If you have the complexity of the protein here, all those shapes represent different protein. Some protein will interact with the solid phase. Some will stay in the solution. You do a separation here. You can either analyze the solution here to get your signal, or you can really how this solid phase do a elution to collect this part. And then we send this one to the LC-MS, mass spectrometry, to get the information. So it's a really lengthy process here. And typically, this process is made by different kind of, let's see, okay. So different kind of resin. So all kind of techniques here, right? So those are the solid phase. They are very expensive, but not very accurate. And the one very important thing here is the current method, they cannot be automated. So you have to do it manually, and you can only handle a few samples at a time. So we think there are three things can really help us to overcome all the challenges we talk about here, right? So one is how we can really do the automation, right? And then second one is how can we really use AI to guide the synthesis of new materials for the extraction, for the interaction with the proteins. And finally, I want to talk about how we can really use AI to do the prediction. So today, I'm going to talk about maybe 3 or 4 very quick case study here. I will focus on the automation part and briefly talk about the AI in the research. So let's go back again, right? Go through that whole process. Typically, if you go to a biolab, either in our own medical school or you go to Quest or or LabCorp or whatever, they try to do this kind of spinning, right? You have the blood coming here, spinning, and then do extraction. It's a really difficult repeating process. So one way to do that is if we can link this kind of operation to an auto-pipetting system or some kind of auto-sampler system, we may be able to solve it. The problem is that the traditional way to do the extraction is always to fix the solid phase to the tips. And when we fix it, the problem is you have all kinds of error coming in here. I don't want to go too much details. The take-home message here is they are not reproducible, and then they vary from one tip to another one. So a few years ago, so I worked with my students and a few colleagues, we designed a new system here. So we based on the available automation. So what we are doing here is a very simple approach. So as I show here, we still use the pipette tips. But in these tips, we have 4 components in it. we design a special freight. So this one is a special materials. They can allow the air and the liquid come in with very low absorption. So there's no interference can come in there. The second part is the resin part. So that's the materials we synthesize, have special properties. The top one is a barrier. We make sure, this one, the air can go through, but the liquid cannot go through. And finally, we have this kind of special 3D printed disperser here to make sure, everything really can suspend it nicely. So let me show something. So that's the whole process, right? The key thing here is this resin, right? This resin is a newly synthesized materials. But that's how the machine. Now we use a Hamilton automation machine here. They can really get everything in it and have about 1 minute at a time, they have this kind of exchange. And the programming is all in the place. It's already all programmed. So we can deal with 96 samples or 384 samples in less than 30 minutes time. So what's the advantage for that one, right? So let me just skip this part, right? The advantage here we talk about is, so the whole process for the whole, we call it conditioning, equilibration, sample, and then washing, elution. Those are the steps for all the extractions. So the extraction step, every single step is controlled by program, can be optimized. So you don't need to individualize for one analyte. You redo everything again. So you can use this robotic system to really optimize everything and give you the best solution here. They can say, oh, I want to have protein A, B, C here. They give you the parameter, and then eventually you get everything. Let me give you one example here, right? So I talked about the phosphoprotein. I will skip the chemistry here, right? The commercially available is the same problem. So we can do something is just give all the parameters in this the table here. We have hundreds of tables, like different kind of resin, different condition, washing speed, whatever, but all of them can be programmed. And then that kind of programming based on the data can come back to reprogram automatically, right? It's a whole self-controlled system. Eventually, you can have the best system. And this is just based on another beauty here is we don't need to design our own system. So there are about 10 different kind of automation systems like Hamilton, Integra, Tecan, Beckman. We can all use them, just change the script, the program a little bit, using our method can plug in. So then, so this is how to set up for the Hamilton system here, right? For each position, you have the reservoir for the tips and something. This is a whole robotic system here. It's a pretty expensive thing. It's about 250K instrument, but it's fully automated, right? So eventually, I'll show some results, right? So that's for phosphoprotein enrichment, very fast, right? 30 minutes, you get about 300 or 100, depending on what kind of tips you're using here. The sample be analyzed. So I want you to see two things. So those two are the control system. It's a commercially used system in LabCrawl or Quest at this moment, right? So using our system, we can have to optimize the system, and you can see the number of the proteins, the phosphoproteins, compared with the protein is the background protein is really high. In other words, you see here, is more than 90% of the protein, they are the phosphoproteins. If you use different other method, right, commercial available method, the benchmark method, so you can see that the phosphoprotein and then the normal protein, they have almost the same number there. In other words, you don't really have any high specificity. And also the error bar in most situations is really high due to the operation. So this is another analysis, right? Just show similar structure here, but you can see if we use the 3, 4, 5, and then 6, 7, 8, those are the special resin for the phosphopeptide, phosphoprotein enrichment. The first two here is a control resin. They're only for normal in the protein enrichment. You can see the specificity is really high here. So this is something I want to show you, is just one of the very simple thing we are doing here based on the design of the materials and then the automation process. So we should be able to do, the really catch the metastable proteins, right? It's very quick. We don't lose any signal. But also, we can have this very quick because we can run 100 samples in 20 minutes, right? So it's really fast, can get signal out. So currently, we have 3 projects using this method now. Like one of them is we're collaborating with a hospital is trying to do how to really profile the colon cancer, the patient. And then we'll collect a sample and then see the phosphoprotein population and the profile and give this kind of prognosis assessment. We try to work with the ICU patients, try to get this therapeutic drug monitoring. The problem for that one is the same thing, right? We started that one before the COVID, and during the COVID, suddenly, all the ICU patients, the pharmacists and then clinicians, they are so busy, right? And then they just drop the ball. And after COVID, it's really hard to go back, to our study. This one, I really see a lot of potential here because, in all the MSCO, typically, about 70 to 80% patients, they come in, you have to flood with all the antibiotics. And then based on the literature, about 40% of the patients, they either overdosage or the underdosing, So that creates a lot of problem. The reason behind it is because, the patients, when they go to the MSCO, typically they have some kind of the history, right? They either have some liver dysfunction or kidney disease, right? When we use a normal, the parameter to give the dosage, we always, not always, in many situations, we'll miss, those, the antibiotics. And so that's something, if we can have such kind of fast analysis, potentially we can see, it can drastically improve the, the prognosis of the ICU patients, especially, for the antibiotics dosage. That's something I really want to, talk to someone, if you are interested in that part, how to use our method to do that. Another one here is we have a special thing, is how to really use the, the PAD, right, the peripheral artery disease. This is something, we collaborate with the clinician. They try to use the, the CT scanning, but they want to combine with the blood samples. The problem they have here is how to really get all this information out. Because now they rely on the ELISA assay and they can only get 20 proteins, but in our method, we can have thousands of proteins come out. So that's something that we're trying to do now. So let me just talk a little bit about the 2 other things we try to do here, right? One is how to really remove the high abundant proteins, and then eventually how to really provide the interesting the information to the doctor here. So that is something we have to rely on the new material synthesis guided by the this data science, and also we try to really add the the how to be more predictable, right? How to really make the sense. Like she talked about there are thousands of proteins, there are so many metabolites, and then all the ions, whatever. How can really put all the data together here? The whole flow here, this is supported by this ADAPT program. We try to have the synthesis initially will have the guidance from the data science based on we call it the MD, the molecular dynamics simulation, and then that can give us the guidance on what kind of materials we need to synthesize. We do the enrichment, and then eventually the analysis. Using the AI and the normal biostatistics to do the analysis, this data come back and feed in to how to really give new kind of the guidance for the synthesis, and then we can optimize the synthesis here to give better results. So it's a whole loop to design the synthesis. Why we are doing that? The reason is because the chemistry for the nanoparticle, there are a lot of them. So how we can know which one is the best? And especially when we talk about thousands of proteins or metabolites in blood, it's really difficult to have this kind of very clear prediction on that kind of chemistry can give this kind of analysis. So the only way to think about it is we can initially give some kind of guidance, right? We talk about the charge density, we talk about the hydrophobicity, blah, blah, right? And then we can talk about the protein or metabolites, what kind of information they have here, right? They have, the size, charge, whatever. Then we make some kind of initial connection. But then, we have to rely on the next step, the synthesis, and then to give the results. And then we do the data analysis to go back. So this just want to show the complexity of the synthesis, right? These are one materials, one kind of size, But then, even when we think of to cover all the charge and then the density and the hyperphobicity, we already go through a lot of things here, right? Many steps, and I don't want to bother with all the chemistry behind it. But that one can only give a few data sets, and then we have to analyze different blood and go back and do that again. And when we go to the blood analysis, it's getting even more messy. Because we have thousands of proteins, right? How can really dig out the importance of each protein, how to make a connection there. So this is something like, we do the analysis, we just do some kind of analysis based on the intensity of the MS result. Then, can do the analysis based on, if I treat, by the way, this is PAD patients, right? So the blue one is an untreated patient, healthy, patient. So healthy sample, sorry, not patient. And then, here is the, sorry for that. So the red one is the patient. So you can see, if you do a normal analysis, you can know, how many proteins we have there. So, but if we dig in the data science, right? So we can do two things here. This circle is the healthy proteins from untreated sample. This one is untreated from the patients. So when I say untreated, means the blood is coming in to the analysis directly without removal all the the high abundant proteins. We can see the difference, but they are really close to each other. It's really difficult. But when we do our enrichment method using the same automation, you can see now is the treated the sample, and then this is treated sample here. You can see the healthy control, and then the patient, they are really far away now. So it's just a very simple thing here. We haven't really optimized everything now. Just initial model can already really amplify the difference and can give us better potentially the diagnosis in the future, or prognosis assessment in the future. So this is something I want to talk about. The AI is we always face the two big problems, including my talk here and Qi's talk, right? The first one is the trade-off between the accuracy of the AI model and then the explainability, right? How you can be accurate, but also you can really explain that one. If you increase the size, big size, you will have more typically more accurate model there. But sometimes because you lose the initial control, the explainability become weak. So that's something how to balance that one. The second thing here is so if you really want to do that one, so how you really to not only to give the decision making from the clinician, right? But also how the clinician can deliver the information based on the AI or machine learning to the patients, right? So for both of them are very critical. So that's something in our thrust, funded by the NSF big proposal. So we are working on is how really can you use the high throughput sample preparation in combined with the sensor, the data fusion, and then some other method, try to really increase the explainability of the AI. So that's the major work we are doing under this program. But because this is a really unique opportunity, I want to talk a little bit beyond my team here. So actually, I started this one about 7 years ago. So that's a part of the project we talk about today. But my major work is really on how to use chemistry and materials for different kind of applications. Like I have been working for more than 10 years to make new materials for just the implantation, how to replace the bone materials. And now one of my students really is in Thailand in the pharmacy school, already use the materials we design here in the dental replacement, right? Maybe Bob can use that one for your surgery, right? That's one part, But in the recent years, we moved to two very important projects also. So I'm really interested in the neurodegenerative disease. I think that's something we are facing every day, and it's become more and more severe because the human in general our health getting improved. So that kind of disease, like Alzheimer or the Parkinson become very severe. We have to find a solution. I have two projects. So this one is to try to synthesize the ganglioside, especially we call it GM1, for the treatment of the Parkinson and Huntington disease. So this is based on something is not a new discovery. It's about 1998. This compound has already been tested in the phase 2 study for the Parkinson disease, but it was stopped by the NIH. The reason, FDA. The The reason is because, that time, all the GM1 was purified, only purified by the brain. And then the trail was based on the cow brain. And then, the FDA worried about the prime or the medical disease, contamination. They had to stop that one. So in the past 3 years, it's pretty exciting. We work with, startup company, Zulia, TRB. TRB is working on the Paulson brain purification, MCS, and the UC Davis. We can synthesize this one in the laboratory with a very, very pure quantity. We don't need to worry about the brain contamination. And also we can change different kind of tail group here. The natural, the GM1, they have different size, but we can really change the size based on our synthesis. So now we try to really find someone to work on this one. So we try to really use this one in clinic, initially for the Huntington disease, but now we try to move to the Parkinson disease treatment. This is the last slide I have here. So we also have another chemistry is based on a small group here, the cyclopropanol. This one, if you are working on the medicinal chemistry, is one of the very popular group. Like if you Google, all the anti-COVID drug, they all have this unit here because it's a good, stable spatial filling. But what we can do is we can have new chemistry here, can open this ring in situ in the cell, and to give the ketone. This ketone can in situ react with the protein. So in other words, this concept of the is a covalent inhibitor. So especially now we're using this one, the cancer-related, the MDM2 protein and the KRAS gene modification. So we have some kind of drug candidate doing that one now, so can drastically, improve the binding affinity. So that's something I'm really excited, and hopefully, someone maybe, faculty here are interested in collaborating for the future. So with that, I want to, thank you again for the talk, and then that's my group, funding agency, and also thank the Wilson family, to this generous support. Thank you.
Wonderful. Thank you both for wonderful talks. My name is Kelly Cannell. I'm the chair of the Biomedical Sciences Department here at the School of Medicine, Greenville, and I will be moderating our Q&A session. I'm sure people in the audience have some questions. Oh, Dr. Rojek has, okay. We're gonna keep it to about 5 minutes for time purposes. So does anyone have a question that they'd like to kick us off with? Yes, great.
Where we should stand? We should stand here?
Yes, that's a great idea. Why don't you both come up?
It's not really a scientific question, but
That's
I'm
okay.
curious as to why it's taken so long to come up with the idea of automating the blood samples, if I have that correct, or the pipetting of them, or the analyzing of them.
So the question is, that's a great question. The reason is it's not as simple as it seems to be. So it took quite some time. About my collaborator, colleague, Bill Brewer started this one for small molecule synthesis, purification about 10 years ago. And then, 15 years ago. Then my student name is Andrew Lee, and me, we work on that one for another many, many years to really make sure it can come to the automation. So there's a lot of nuance, like every small thing here, it seems very easy, but you need hundreds or thousands of experiments to optimize that one. Yeah. So it's not as easy as sound, Yeah. But now, it's really good. But now we are working on is we have the techniques we can transfer to all kind of automation system now. So we have 10 different kind of automation. Almost all the membrane, the system we can use that one now. Yeah. But thank you for the question.
And I have, you have one. Okay, go ahead.
Hi, great talks, thank you. I had a question about the digital twin research. Many of us here are educators, and also we're interested in lifestyle medicine here, which has a lot to do with behavior. Do you think there is any potential use for digital twin to either predict the effectiveness of different educational interventions with learners or different behavioral interventions, such as for lifestyle medicine on health outcomes?
Yes, absolutely. Actually, we have a consortium right now. Is called the Digital Twin for Health Society. We just registered the nonprofit a few weeks ago, and we are electing the president and so on and so forth. And we submitted a proposal last year to NIH. Actually, we are one of the module in the proposal is to pay attention to, for example, mental health for elderly. So in that component, there are some educational, component. I think almost 50% of educational components is to educate the elderly using digital twin as a technology platform. So absolutely, we can develop a digital twin for educational purposes, and basically use this as another way to implement AI in that capacity. So yes, but for us, we are not working in that area. We are more like to work with the people in the clinical field, and so we don't have any first-hand experience with that kind of a digital twin yet.
Wonderful. Well, one of my questions was, could you use, when you're faced with a limited data set, is there any value in using some of that data set to create permutations and artificially grow your data set for further training? Is that something that is considered? I know sometimes we do that in the learning space when we have limited numbers of learners taking assessments. We can kind of create artificial rounds of data with that to look at the robustness of our assessments. And I wondered if that's something that could be done here as well.
Yes, absolutely. One very hot tool in the last couple of years is called the diffusion model. And the diffusion model basically is a tool exactly try to implement what you said. For example, you have a limited data set. However, if you can learn the probability behind the data set, and then you use that probability to regenerate the new data set, that's what the diffusion model is doing. And it turns out that there are many new tools available for diffusion models. You can start out with the sparse data, and then you can create a very coarse-grained probability distribution. And then along the way, you can refine that. If you have a new data coming in next year, and then you add to your existing ground truth data, and then you refine that probability. But over the years, your regenerated data will become more and more similar to the original data. So this technology is still evolving, but I think is dominating the entire field at this point. That's what we are doing in our models, using imputation models. So we tested many different models. Diffusion gives the smallest error at this point.
Great, it's a good idea. Okay, wonderful. Can I ask one more? Sorry. Okay. So sorry, I'm never going to get invited to do the Q&A again. But I'm actually, my training is as a cancer biology pharmacologist. So I was curious when I was listening to your talk about whether you could pick up proteins that have mutations that might alter their chemical properties, like their lipophilicity. If you have a mutation state on the phosphoprotein that mimics phosphorylation sites, would this be a useful tool for that? Because there could be a real niche market for that, but in colon cancer in particular.
I think that's a great question. That's the motivation behind our study here. So for the static proteome, it's pretty easy. But for that kind of mutation or changes, we call the spatial temporal effect, right? It's a very, very popular field. So that's exactly, the fast reproducible data is critical for that kind of analysis. But that's a great question, yeah. It will be the direction.
Wonderful. All right. Well, thank you both very much for very invigorating talks. And I think we're all inspired by this evening. So thank you. And now we will move into the rotunda for refreshments. Oh.